Hi students, welcome to HSC Biology and Module 7, Infectious Disease. This is the last in our small series, it's video number 21, and we're looking at genetic engineering. So in this little series where we've been investigating some of the procedures that can be used to prevent the spread of disease, we're going to look at a final one now, which is genetic engineering. So in this case, and we've kind of done things a little bit out of order, so we haven't looked at genetic change yet, which is Module 6, and we will be having a look at that in a little bit more detail, and obviously that'll provide a lot more background to this particular little part of infectious disease, but you should be able to define the term transgenic organism. You should be able to describe how organisms can be genetically modified, particularly in relation to disease resistance, and perhaps to be able to evaluate the impact of ethics in genetically modified organism biotechnology. So genetic engineering is just the altering of the genetic composition of an organism in some artificial way. So we've now gained a great deal of understanding of the structure of DNA, of the sequence of bases. We know the relationship between the base sequence and the protein or the sequence of amino acids that are coded for. And so therefore we can actually make changes to that code if we know exactly what the consequence of those changes are going to be. We can also splice little genes out from one organism and glue them, stick them into another one, and to determine whether or not that organism will be able to express the genes that it didn't initially have. This is a technology that's been applied to increase disease resistance, but to try and do it in a more natural way, despite the artificial nature of the structure, it means we're not using chemicals in pesticides, herbicides, fungicides, all of those sorts of things. We're trying to create some sort of inbuilt resistance within the organism itself. We hope that the use of genetic engineering will also help to control the spread of disease throughout the population, and to define that first step that we wanted to look at on our little success criteria was that if we have organisms that contain genes from other organisms that have been inserted into their own genetic material, well, this is our definition of transgenic. So trans just means across. So genes across species. So if you have genes in your body from another organism, then you would be transgenic. That obviously doesn't happen a lot with people, but it does happen a lot with some of our agricultural crops and also some different types of animals. So we're going to have a look at this in a little more detail. The GMOs are one of these areas where there are some ethics associated, where there's labeling, and where also there is legislation, which is not the same in all places in regard to labeling or the use even of genetically modified organisms. But genetic modification has occurred in a number of very important agricultural crops and livestock. So just a few examples, you can see them kind of in the background of this slide, but also in my list here, cotton plants to protect them from insect attack. Likewise with corns, a lot of genetic engineering has happened with corns because it's such a big crop, not just as a food crop for humans, but also as a feedstock and for multi-purposes corn. Beetles, potatoes we've already had a look at, and beetles can be one problem with potatoes, but we've also looked at the fact that viruses can attack them as well. We've looked at modifications to cattle, specifically trying to deal with resistance to mastitis or resistance to tick infestations. But we've seen, and I'll show you in a second on the next slide, how cows have actually been used to produce human antibodies, which can then be cultivated and used. And we've also seen chickens be able to produce human proteins. So these are examples, in most cases, of transgenics, where we've introduced a gene for a particular protein, which might be a human antibody or might be another type of human protein. We've introduced that into an organism at the very early stage. As its cells have reproduced, that particular gene has reproduced. And of course, we also need to be able to trigger it. We also need to be able to turn that gene on so that it will produce the particular protein that it codes for. And this, as I say, has happened in a number of different types of different organisms. And you can see from the some of the pictures in the background there are plenty more examples. There's lots of examples of genetically modified organisms for various purposes in various different parts of the world. One specific application of this kind of idea of genetic engineering and transgenics is in the application of human antibodies. And we know that when we're looking at our vaccines, we know that there were ways we could use both passive and active ways to acquire some level of resistance against specific pathogens. So again, these are part of our specific response. And we said one of the ways to do that is maybe to introduce a vaccine which has an attenuated version or some sort of a toxoid or something that's come from a particular pathogen that triggers our immune system to produce a response. But there's passive ways as well of doing that. So the passive ways can include things like in utero transfer of antibodies between the mother and the baby, but also this injection, this sort of artificial, if you like, a vaccine that can actually deliver particular antibodies to an individual. So it's a vaccine in a slightly different form. It's not necessarily stimulating the body to produce its own antibodies. What it's doing is just giving a little bulk load so that when the pathogen is encountered and the body does start to produce a response, there are already antibodies in the system that are able to counteract that pathogen. And you can see we've had some success in trying to be able to produce human polyclonal antibodies amongst cattle and to be able to recover those for pharmaceutical use. So these are just some of the examples of genetic engineering. There's a number of different ways now. You should have a nice list of the different ways in which we look at seeking to limit the spread of infectious disease. And genetic engineering is just the last in that series. So we'll pull them all together and put them in a table, which I think is a good way of summarizing this sort of information. But we'll do that in class. Thanks for watching.